Now we will turn our attention to aligning RNA-seq data. Instead of sequencing DNA fragments, mRNA fragments are sequenced. This allows the investigator to study the transcriptome of cells of interest, which provides information regarding protein synthesis at the time the samples were prepared. Tag sequences are aligned with a reference genome, but because the tags are RNA, they align to a large extent only to the exons in the genome, not to the introns and not to regions of the genome that do not code for genes. Image analysis, base calling, error estimation, and quality thresholds are performed using the sequencing machine's proprietary software. In the case of Illumina, the HiSeq controller software. Thereafter, other preprocessing steps can be applied using publicly available software. You may recall that as part of the assays process, adapters are ligated to the sample fragments to be sequenced, and the adapter plays a role in sticking the fragment to the surface of the flow cell and in the bridge amplification process. However, we do not want to align a sequence that includes adapters because the adapters would not be in the reference genome. CutAdapt is a publicly available software tool used for adapter trimming. Example syntax is given here, where the CutAdapt program is called. The parameter -a means what follows is the adapter sequence, so of course next the adapter sequence is provided. Then the original gzipped FASTQ file is provided, which is piped to this output FASTQ file. Other programs can be used for trimming adapter sequences, including these. RNA-seq data may contain a substantial amount of ribosomal RNA, transfer RNA, and mitochondrial RNA. These can be filtered out in the bioinformatics pipeline by simply using a contaminant file of rRNA, tRNA, and mtDNA sequences for the target species to pre-filter all sequence reads by alignment before mapping the reads to the genome or splice junction database. This can be accomplished by aligning your FASTQ files to a reference contaminome database that includes the unwanted sequences. You would need to construct this reference database using publicly available resources as HSA contaminome, for example. Then when you call the aligner, instead of keeping aligned reads, you use an option that writes the reads that did not align to the contaminome reference database. That leaves you with decontaminated reads. Bow ties -un and -un-conc writes unpaired and paired reads respectively that did not align to the contaminome reference database. A commonly used approach when RNA sequencing assays were first introduced was Illumina's Cassava RNA-seq pipeline, which uses the Efficient Large-Scale Alignment of Nucleotide Databases, or ELAND, for sequence alignment. First, ELAND determines the positions in the genome to which read substrings, which are seeds of 16 to 32 base pairs, match with a maximum of 2 errors in a process called single seed alignment. The best alignment for that seed is picked using alignment scores. Reads that do not align are flagged, and multi-seed alignment is performed on those flagged reads. In multi-seed alignment, up to 4 seeds per read are used, which are multiple contiguous, non-overlapping sections of 16 to 32 base pairs within a read. Because up to 4 seeds are currently allowed, that means that only up to 3 32 base pair seeds are possible for a 100 base pair read. Then, for each seed, an alignment score is computed by taking the p-value of the candidate seeds and dividing it by the sum of the p-values of all of the candidates, which is then converted using a Phred scale. The highest Phred value indicates the best candidate. The Illumina method calculates the gene expression value as the sum of the total number of bases from all reads that align to exons and genes. The number of bases that fall into exonic regions of each gene is summed to obtain gene level counts, which is then normalized by reads per kilobase per million, or RPKM. Note, normalization will be discussed in Module 5. The Cassava software also reports the number of reads that map to each exon, so expression is reported by both gene and exon. Later, the Top Hat software became commonly used. The Top Hat developers recognized that Bowtie is a useful and efficient program for sequence alignment. However, Bowtie is not suitable for RNA sequence alignment because introns are spliced out of the messenger RNAs, creating large gaps between a read and the genome. Top Hat was developed to address this limitation. Top Hat uses Bowtie as an alignment engine to align reads to a reference transcriptome, then breaks up reads that could not be aligned into smaller segments, and then aligns these previously unmappable reads to the full genome using Bowtie. Often, these smaller segments, when processed independently, will align to the genome. When several of a read's segments align to a genome far apart, for example, between 100 base pairs and several hundred kilobases from one another, Top Hat infers that the reads span a splice junction and estimates where that junction's splice sites are. Therefore, it identifies splice junctions de novo from gaps in the alignment of reads to the genome. After Top Hat alignment, cufflinks is then used to quantify the sum of all reads that map to a gene across exons and all intron splice junctions. Accurate de novo discovery of splice junctions requires very deep sequencing coverage because each splice site must be covered by multiple reads with adequate overlap with the exons on both sides to allow for alignment. Cufflinks was designed to work with Bowtie and Top Hat sequence alignment programs, but it can use any file of RNA-seq reads aligned to a reference genome in SAM or BAM format, for example, such as those produced by the BWA aligner or Illumina's Elon Cassava package. The output from Top Hat is a SAM file. Cufflinks works best with paired end reads where it can calculate the size of RNA fragments used for sequencing, which in turn can provide a great deal of additional data regarding splice isoforms. Cufflinks requires deep coverage of the transcriptome without extensive 3' bias in order to work well. Other alignment software includes spliced transcripts alignment to a reference, or STAR. Again, mapping reads derived from non-contiguous genomic regions, such as mRNAs, whereby exonic regions are spliced together and introns are removed, is a challenge. Other RNA-seq aligners work primarily with relatively short reads, that is, less than 200 bases, whereas STAR can align long reads, which are even several kilobases, and it has high mapping speed. STAR includes two major steps: a seed searching step and a clustering/stitching/scoring step. In the STAR seeds searching step, the longest substring of a read that matches exactly one or more substrings of the reference genome where the maximum mappable length is recorded. In the clustering/stitching/scoring step, STAR builds alignments of the entire read by stitching together all the seeds that were aligned to the genome in the first step. The seeds are then clustered together by proximity to a selected set of anchor seeds, and all the seeds that map within the user-defined genomic windows around anchors are stitched together, assuming a local linear transcription model. The stitching is guided by a local alignment scoring scheme with user-defined scores for matching and penalties for mismatches and gaps. The stitch combination with the highest score is chosen as the best alignment of a read. The output from STAR is a SAM file. A program like HT-seq is then used to count reads per gene or per exon. Another program, Callisto, uses pseudo-alignment to quantify abundances of transcripts from bulk and single-cell RNA-seq data. Pseudo-alignment focuses on identifying the transcripts from which the reads could have originated and does not try to pinpoint exactly how the reads align to the transcriptome. This is accomplished by a technique of hashing k-mers with the transcriptome de Bruijn graph. Because reads have errors, k-mers with errors are ignored. The abundance data from Callisto is a tab-separated file that includes target ID, which is the transcript ID that the reads map to, length, estimated counts, and transcripts per million. Other RNA-seq alignment software includes GeoSnap, RUM, MapSplice, among others. The issue is that if you grab some old RNA-seq data files that were analyzed with Cassava and then realign them with Top Hat and then with STAR, you will have 3 BAM files for the same sequence data. Unfortunately, choice of software for alignment and expression counting do influence final results. Best practices evolve over time, which means that conclusions drawn from earlier software methods may deserve re-examination. Therefore, best practices include storing your raw data as FASTQ files for the long term. In summary, when pre-processing RNA-seq data, be sure you understand the platform and protocol used to produce the RNA-seq data, as tasks such as adapter trimming and decisions based on single versus paired end sequencing alignment require different parameters to be passed to the software, and Fred's quality scoring differs. Pre-processing steps include trimming adapters or asking whether adapters have already been trimmed in the data that you have been provided. If you are not interested in certain species of RNAs, such as ribosomal RNAs, mitochondrial RNAs, or transfer RNAs, then decontaminate your FASTQ files prior to alignment. Then map reads to the genome and count reads per gene and/or per exon. In upcoming modules, we will discuss normalization and statistical analysis. While we will be using R and bioconductor packages, the methods that I have described are largely publicly available and can be run from a command line prompt, with exception of Illumina's proprietary methods. Additionally, Galaxy is an open-source web-based platform that can be used for pre-processing next-generation sequencing data, including RNA-seq data. The Genome Analysis Toolkit is a structured software library produced by the Broad Institute that contains tools for many types of analysis of next-generation sequencing data, with a particular emphasis on single nucleotide variant detection for human resequencing projects. GATK provides an extension of the single nucleotide variant tools in SAM tools, implementing a pipeline that includes mapping, quality score recalibration, multiple sequence alignment, and SNP calling. It is important to have an understanding of alignment methods. However, most often, biostatisticians are provided with data that have been already aligned. Because of the high computational cost of aligning RNA sequencing data, in this course, we will not be aligning large data sets of RNA-seq data.